Yeah, we only have biomarkers. 40.
I'm Giorgio Ziola, the Director of Philanthropy for the ADDF, and I'm happy to be back in Washington, D.C. with you all today for this great event. In a few moments, we're going to hear from the experts at the symposium, and we're going to enjoy a terrific lunch and fashion show afterwards. And this Great Ladies Luncheon was created by Elise and Mark Lefkowitz to honor in memory of a very great lady, Estelle Gelman. And Elise wanted to continue her mother's incredible legacy of philanthropy and fighting to end this disease that took her from us. On behalf of everyone at the ADDF, I want to thank Elise and Mark for all they do to put this event on every year, and it's wonderful, better and better each year, it seems. And I also want to thank our longtime partners, Neiman Marcus, and the generosity of all our sponsors, Carolina Herrera, Estee Lauder Companies, and Lilly. And of course, I want to thank each of you for being here and for the generous support that you've been lending to this event over the years. It's been an amazing year for the ADDF. We've marked some incredible milestones. And since last year's luncheon, we announced an incredible new partnership with Bill Gates, the Dolby family, the Charles and Helen Schwab Foundation, among others, to create something called the Diagnostics Accelerator, which advances the development of novel biomarkers for Alzheimer's. Most recently, the ADDF welcomed some additional partners to the Diagnostics Accelerator, including the Association for Frontotemporal Dementia, and the latest to join the coalition is Jeff and Mackenzie Bezos. And that brings the current program funding for the Diagnostics Accelerator to $50 million. So we're very optimistic about the changes that will come for this and what this means for the entire field. Dr. Fillit and our esteemed panel will explain today what biomarkers are and tell you a little bit more about that and why they're so important to what we do. And we'll also hear more about what we can do now to prevent Alzheimer's, which is something I'm sure everyone in the room is interested in hearing. So I'm thrilled, without further ado, thank you. A familiar face, highly respected TV journalist. Andrea is the NBC News Chief Foreign Affairs Correspondent and host of MSNBC's Andrea Mitchell Reports. Please join me in extending a warm welcome. Thank you.
Thank you all so much for being here. Thank you for the kind introduction. I just want to say, it's not as though there isn't a lot happening in Washington right now, including today, as the hearing is convening with the Attorney General. And since my main job is foreign affairs, we don't know what's happening in Venezuela, and we may be at a tipping point there. I'm supposed to be interviewing the National Security Advisor at 12:30, John Bolton. So again, I have to apologize. It's not for lack of wanting to be here and listen to what Dr. Fillit is telling us about all of these wonderful achievements and all of your colleagues here. But I have to go to work for my real job. But I'm here for a lot of reasons. First of all, Elise and Mark Lefkowitz and what they've done in honor of Estelle Gelman and her incredible philanthropy and the wonderful human being she was, and this legacy goes on. Second of all, my friend Leonard Lauder, who has been so instrumental in honor of his mother, Estee, to what the Lauder family has done here. And my own engagement with the Lauders at the University of Pennsylvania, and also at Memorial Sloan Kettering, where I've been a patient. So for all of these reasons, friendship and love and affection, my own mother had suffered, and we lost her it's about 7 years ago, and watching what she went through, I can identify so strongly with all of the patients, the caregivers, often the children, often the women, the daughters, more importantly than some of their siblings. So this is a family, a national and a global concern. And I just want to pay tribute to the medical teams here, the researchers who are doing such extraordinary work. The Alzheimer's Drug Discovery Foundation is unique in the way this benefit and all of its fundraising works, and all of the money going into the research. Alzheimer's, as you know, is a growing public health burden, and that's not just here in the US, it's a global concern. especially as we baby boomers age. It's becoming an increasing, really an epidemic, even though obviously the causes don't relate to epidemiology, but what we have is an aging population, and we're not caring for our elderly, especially those who don't have access to the best medical care. So we have 5.7 million estimated Americans with Alzheimer's, 50 million people around the world, and they expect that that number will triple to nearly 14 million in the US by 2060 and 150 million around the world, unless we do something to stop the progression, and that's where the medical team, the researchers, come into play. It's a huge burden on society, so this is an economic drag on our country as we all age. It's a tremendous physical and emotional toll on patients and caregivers and families alike. I think there is increased awareness with more reporting, with some of the work that we all do in my profession, but not nearly enough on the real impact on our economy, on people, on the society, on families. And I think the best approach, clearly, is to focus on research, which is exactly what ADDF does. The good news is that this organization that you are so generously supporting is absolutely on top of this, working on the cutting edge, the best and brightest researchers around the world getting us closer to a cure. It is the only charity, as I just said, solely focused on finding drugs to prevent and treat Alzheimer's disease. Nearly 1 in 4 of every treatment now in clinical trials for Alzheimer's has received support from the ADDF. And today we're really privileged to hear exactly what is going on in this extraordinary research from the scientists who are deeply invested and in charge of all of these discoveries. Our panel discussion, Biomarkers to Accelerate Drug Development for Alzheimer's Disease, will be led by Dr. Howard Fillit, who we are so grateful to, the founding executive director, the chief science officer of the Alzheimer's Drug Discovery Foundation. Dr. Fillit, as many of you know, is internationally recognized as a leading expert in Alzheimer's disease. As a geriatrician and neuroscientist, he has been involved in Alzheimer's research and care for almost 40 years and has led the ADDF since it was founded in 1998. It's incredible that it has been so long that you have been leading the way at the ADDF. And he's joined today, we are honored to say, by Drs. Eric Siemers of the Siemers Integration, Marilyn Albert of Johns Hopkins School of Medicine, and Richard Isaacson of Weill Cornell Medicine. We'll learn today about the role of biomarkers in early detection and accurate diagnosis, in monitoring disease progression, advancing clinical trials, and prevention. And most importantly, in addition to learning so much, you're all going to have a chance to ask these extraordinary scientists questions and interact with them and get the benefit of that. So it is my great pleasure to introduce Dr. Howard Fillit. And Dr. Fillit, I know you're on the cutting edge of all this research. If you can find an answer to congressional hearings and what brain damage, my obsession with following these hearings and then watching and reading and staying up all night, getting on top of it. If you can help me with that at some point, you'll really be a hero. Thank you all so very much, and have a wonderful day.
Thank you very much, Andrea, for being here, for working with us all these years, and for your wonderful introduction. I do have a suggestion as a geriatrician and neuroscientist. I think the first thing we should do is a mental status test on everyone. I would think that would help. Okay, thank you. Well, thanks to all of you for being here today. We've constructed this panel to give you some idea and some information about the role of biomarkers. I think the importance of it is only strongly illustrated by the fact that Bill Gates, Jeff and Mackenzie Bezos, David Dolby, the Schwab family, and others, including Leonard Lauder, have invested heavily in promoting this. And the paradigm I would give you is the role that blood tests for cholesterol the way that blood tests for cholesterol just revolutionized completely the field of heart disease in terms of diagnosis and clinical trials. But it took 35 years for going from recognizing cholesterol as a risk factor until the first statins came to market. Today's panel is going to address this issue. Dr. Siemers, the first person on my left, has had a long career in drug development at Eli Lilly and is a leader in the field of clinical trials and is going to talk to us about the current state of drugs in clinical trials today. Dr. Marilyn S, has been in the field for many decades like ourselves and will at Johns Hopkins and will tell us about the role of biomarkers in diagnosis and their use in clinical trials. And Dr. Isaacson has become a world leader in prevention, a very forward-thinking physician, established a tremendous clinic for prevention at Weill Cornell, and he's going to tell us about what brain health is and how we can all achieve that. I'll be moderating the session. I'll ask the panel a couple of questions. Once everyone's had a chance to speak, we'll open it up for your questions. Let me just give you some preliminary remarks. I've been doing this about 40 years. Saw my first patient with Alzheimer's disease in 1980. At that time, I'd been involved in medicine about 10 years, and I had never heard of Alzheimer's disease. And for those of you who were around back in the 70s, you probably never heard of Alzheimer's disease either. So the field was relatively young, and compared to cancer, the first clinical trials in cancer were done 250 years ago. The Imperial Cancer Trust in England was started for research in 1902. We didn't start our research until the late 70s, early 80s. But I think we've come a tremendously long way and rapidly in these last 35, 40 years. And to me, just in my professional lifetime, it's just amazing how far we've come. Today, we have a much better understanding of the disease. I think we know a tremendous amount about the biology of the disease. We have FDA-approved diagnostics at this time. In the old days, when I wanted to make a diagnosis of Alzheimer's disease, I could only do it on a clinical basis, a brain biopsy, or an autopsy. And I can tell you I wasn't doing a lot of autopsies to make a diagnosis on my patients. Today, I can write a prescription for a radiology exam down the block, a PET scan, although it's not paid for by Medicare yet. I think it will be. And I can make a diagnosis of Alzheimer's disease with about 98% or more certainty. And we've learned a tremendous amount from that test. We know now that, as was mentioned, by doing the test, the brain scan and spinal fluid test that we have, these are biomarkers currently available. We see that Alzheimer's actually is a disease of middle age. It starts in our 40s and 50s and perhaps 60s and progresses before there are symptoms. But beyond the diagnostic tests that we have for early detection, they play a critical role in clinical trials for enrolling people into trials and then for using the test as monitoring. And Dr. Siemers is going to tell us about some of the way that's been used. But the big picture is that going from a time when we had no drugs in clinical trials, and I ran one of the first ones back in the early 1980s. There were no drugs in clinical trials and no drugs on the market. Today we have over 100 drugs in clinical trials, and about 25% of them are on this amyloid target that you've heard a lot of failures about. Dr. Siemers will give us his interpretation of where we are and how that's contributed to our knowledge. But 75% of the drugs in clinical trials today are not amyloid, and we have an increasing number of clinical trials that are on novel targets like inflammation and neuroprotection and other targets like big words, autophagy and epigenetics, which I don't have time to go into. And finally, what we know about today that we didn't know about 40 years ago, or really even 10 years ago, is that with research, with very rigorous research going on around the world, that it really is starting to look like we can prevent Alzheimer's disease. And Dr. Isaacson is a leader in that field, and he's going to tell us how he's taken prevention research and translated it into clinical practice. And the soundbite that is probably most important to remember and is really seeming to be true from clinical research is that everything that you think of to prevent heart disease and you're aware of will help you to achieve brain health in addition to certain other factors like continuing social and occupational engagement and hearing loss and things like that. But it's possible to delay the onset of Alzheimer's disease. Biomarkers are going to help. We're going to have digital tools. Our diagnostics accelerator that we're doing with Bill Gates is also going to not only, the goal of the diagnostic accelerator, the immediate goal, is to commercialize a blood test for Alzheimer's disease. Imagine if you could go to your primary care doctor and get a blood test, and within 24 hours or so, the doctor could tell you whether or not you have Alzheimer's, whether that problem that you have with your memory could be due to Alzheimer's, or even if you don't have a problem with your memory and just want to be able to know what your risk is. And I honestly think that with the blood tests that we're seeing in development today, that in 3 or 4, maybe 5 years, we'll have a blood test on the market for Alzheimer's disease. And just as cholesterol, like I said before, revolutionized heart disease, I think the blood test will revolutionize both clinical care and research in Alzheimer's disease. We're also going after digital biomarkers. I've been told amazing things about how these watches work, and we're going to have digital biomarkers. We already do for Alzheimer's, and we're going to be able to really improve care and clinical research by the use of digital biomarkers. So for me, 40 years into this, everybody's got a real nihilistic point of view about what's going on. We've heard about all these failures. I got to tell you, I have never been, in my entire career, 40 years, my professional lifetime, more excited about the work we're doing than I am right here today. We've got blood tests we've got diagnostic tests approved by the FDA. We've got over 100 clinical trials, many of them on different targets that could potentially help. And we know a lot about prevention, and the future is very bright. I'd like to introduce now Dr. Eric Siemers. Eric, like myself, has been involved in the field for many decades and spent a long time, I think over 25 years, at Eli Lilly, where he was very much in charge of clinical development at Eli Lilly, and played a tremendous role in our field in helping us to learn how to do clinical trials and then conducting those. And he's going to tell us about his remarks on clinical trials in Alzheimer's disease. So, Eric, please.
All right. Thank you. Well, thanks very much for that nice introduction. I did spend, actually I started the first part of my career at Indiana University and then was at Lilly for a long stretch, and then just recently started a consulting business. And so that's been interesting in that I've been able to have this viewpoint not of just one pharmaceutical company, but looking kind of a little bit across the board. So to get to some of the big picture issues, I mean, I could immediately dive into the nerd weeds here, but you probably don't want to hear that. But you probably have heard that there have been a couple of recent disappointments in terms of big phase 3 trials for Alzheimer's disease. One of those that's been the most recent is a drug called aducanumab that Biogen was developing. Those phase 3 studies were unexpectedly stopped early for futility. It wasn't going to work. And then prior to that, earlier this year, there was a drug called crenezumab from Roche. That one was also stopped early. That one maybe wasn't quite as unexpected, but those things are always really disappointing for everybody who's hoping for better treatments. And on the other hand, there have been positive things too. Last fall, there was another drug called BAN2401 that had some phase 2 results that looked positive. So it's a little bit of a roller coaster. And I think the analogy of cholesterol is a good one. It takes a while to work through these things. It's a little bit like the Apollo program. It wasn't Apollo 1 that went to the moon, it was Apollo 11. I think right now we're kind of Apollo 6 or 7, somewhere in there. We don't know when we're going to get to Apollo 11, but hopefully it'll be sooner instead of later. But to get into a little bit of the specifics, for those of you who may read this, and a lot of it comes across in the media, especially these 2 most recent drugs that were negative in phase 3 trials, they were based on what's broadly called the amyloid hypothesis. And you'll read, especially some of the investment people will say the amyloid hypothesis is dead. In my opinion, the rumors of its death are greatly exaggerated. So I don't think that's really the case. The pathology of Alzheimer's, as you heard, starts probably at least 15 or 20 years before you get any symptoms at all. So your brain has this cognitive reserve, and it takes a while to sort of burn through that reserve, even though you have the pathology. And the first part of the pathology is this amyloid. Now, amyloid, to give you a couple details, there's a little protein called A beta. It's kind of sticky, and when it globs together, it's called amyloid plaques. There are some other species in there, and the point is, broadly, is that the field hasn't really attacked all the species of A beta or amyloid, and in particular, the ones that most people now think are the ones that kill the neurons really haven't been targeted very adequately. So again, I think the rumors of the death of the amyloid hypothesis are greatly exaggerated. In terms of other targets, though, that you heard about, one of the exciting new targets is another protein called tau. Now, it's the second thing besides amyloid that's the key part of the pathology in Alzheimer's disease. And there's now clinical trials that are ongoing, relatively early phase, but clinical trials that are targeting this other protein called tau. Now, again, in my opinion, one of the things everybody has to be aware of is that in the early days of doing these things, there will be disappointments, and that doesn't mean that you should just stop and give up. And especially in these early phase trials, you're not going to get a definite answer. That'll take a few years, but it's a very good target and something that needs to be addressed, and it's being addressed. So that's an exciting advancement in the field because that's relatively recent. Now, the third thing that I would actually focus on are treatments that are there's maybe one that's entered the clinic, but it's newer. And it's based on your immune system. So there's always been this suggestion in the field that your immune system, maybe there's too much inflammation, that's bad for the brain cells, that's why they die. It's been a little bit difficult to sort that out, but more recent genetic studies have said, yeah, that's the other thing. In fact, all the genetics either point to amyloid or the immune system. So that's the third thing that's just now starting to enter clinical trials that I think actually has a lot of promise. There are some other mechanisms out there that probably too complicated to get into, but I think those are the three main things: amyloid and A beta related, tau, and then these newer immune-related therapies. Now, with that being the case, with these different ways of going about it, one of the things that I think most people in the field agree with is that it's going to be combination therapies in the future. There's probably not going to be one magic bullet that will cure your Alzheimer's disease or prevent your Alzheimer's disease. So in the future, we actually expect that two or more of these compounds might be used in combination with each other, just like you might use more than one thing to control your blood pressure or your diabetes. It's not uncommon in medicine. So that's really looking forward into the future. In the shorter term, you may hear about clinical trials that are actually designed to look at not just one investigational drug versus placebo, but two different ones, and there are study designs that can account for that. So I think that's the really longer view, promising aspect of this. Again, to get back to sort of Howard's closing point, you get some disappointing press sometimes, and you get people talking about, well, this didn't work, let's just quit. I don't feel that way at all. I feel like we're sorting out, even of the amyloid-related targets, which ones you really have to go after. There's these new targets that are starting to be addressed, and we have a much better understanding of the biology of the disease and how to use biomarkers both as a diagnostic and to help the actual drug development process. So I think that's a good segue then to the next topic. Or did you want to? Yeah.
So Eric, you've been involved in clinical trials for Alzheimer's disease for decades. And I think we have to applaud the pharma industry for spending tens of billions of dollars investigating drugs for probably 25, 30 years. But what have we learned about how to do clinical trials in Alzheimer's disease? I remember in the old days when I did it, when I look back at the patients I enrolled, for example, I think I have no idea who those people were. And Lilly did a sentinel study with solanezumab to show that about 30% of people that were being entered into trials actually didn't even have Alzheimer's disease and didn't even have the target we were looking for, which was the beta amyloid. And the PET scan changed that world, which you guys own that one. But in what ways can you tell us that we've learned how to do better clinical trials so we're going to get better results?
So that's one of the real advancements in the field, and maybe just to make one quick point, I don't want to get into drug pricing in the pharmaceutical industry and that sort of thing, but I will say that Lilly spent over $3 billion on Alzheimer's research, and they've never made a penny from that. So it's a tough business, especially with a disease like Alzheimer's. It's a tough nut to crack. So in terms of what we've learned in terms of clinical trials, it is kind of amazing when you think back. When we first started to do, or when I first became involved in trials at Lilly, we didn't have the diagnostics, and so now we know in hindsight that maybe 25–30% of people actually didn't even have Alzheimer's disease. They had another cause of their dementia. And then if you get into a little bit more of the technical details, one of the things that we spent a lot of time with is using biomarkers to figure out what dose we needed. And in terms of drug development, finding the right dose is, you may think it's easy, it's actually the hardest thing. And so in order to use these biomarkers correctly, in order to improve our clinical trial designs, we applied these biomarkers to help us understand that the dose of the medicine that we were testing was actually relevant, and it was doing what we wanted it to do. It's called target engagement, is what we call it in the field. So it's kind of these two aspects. One is better diagnostics, knowing that we have the right people in the study. Second thing is finding better ways to actually know that the drug is doing something. And then actually the third thing that the field has learned along the way recently is this idea that the pathology starts 15 to 20 years before you develop symptoms. And so there may be some therapies that if you've developed symptoms, it may be too late, but they may be very good for preventative treatments. So that's, I guess the third thing the field has really learned is how long it is after the pathology starts it is until you really get symptoms.
So Eric, we know that one phase 3 clinical trial costs $400 million in Alzheimer's disease, and you need 2, 3, 4 clinical trials to get an FDA approval. So it's a tremendous risk. And as I understand it, about half the cost of a clinical phase 3 trial is enrollment. It's just trying to find the patients, screen them, make sure they have the disease, and get them into the study. And we know that, I think also it's correct to say that less than 5% of people out there with Alzheimer's disease actually enroll in clinical trials. So how are we going to make it better to get more people aware of clinical trials, enrolled in clinical trials? How can we make screening and enrollment more efficient so the cost of clinical trials can be mitigated a bit?
Yeah, no, that's a great question. And so, as I've said many times, the rate limiting step for getting a really potent treatment for Alzheimer's disease is enrollment in clinical trials. You're not going to have any drugs FDA approved without the clinical trials to prove that they're safe and effective. And it's unfortunate that most people don't participate. Now, I can give you an example. My father-in-law now has kind of mild to moderate Alzheimer's disease. He was in Florida when his symptoms started. I was up in Indianapolis, and it was too much work. We couldn't get him into a trial, and this is my father-in-law. But it's really important, and as the field moves earlier, there are now studies that actually involve people who are cognitively normal. So you talked about the screen fail rate. When you screen people for trials, not everybody qualifies. And so if you're doing one of these trials where people are cognitively normal, you're looking for people who on the imaging study that you heard about have these amyloid plaques. Well, for people age 65 and over, it's a little frightening that 30% of people roughly have the amyloid plaques even when you're cognitively normal. So that's frightening. But on the flip side, 70% of people don't have the plaques. So that's good, but it makes your trials difficult to enroll and more expensive. Typically for trials, even for people who have symptoms, the length of time it takes you to enroll everyone into the trial is about the same length of time as the actual trial itself. So if you could shorten that enrollment period by having more people get into trials, it would be terrific. There are some registries that are around that are starting. That's one aspect that maybe we can come back to. But I think mostly it's awareness, and I think groups like this are the people who have the most interest and are aware, but just realize that participation in trials is actually the rate-limiting step.
Thank you, Eric. Our next panelist is Dr. Marilyn S. and she's a professor of neurology and director of the Division of Cognitive Neuroscience at the Department of Neurology at Johns Hopkins here. And she's also the director of the Johns Hopkins Alzheimer's Disease Research Center. Her research is focused on understanding the cognitive brain changes associated with aging and Alzheimer's using biomarkers and imaging on spinal fluid and genetics to better understand the earlier phases. And it's my pleasure to introduce Dr. Albert, who's going to tell us a little bit about what biomarkers are and how we can use them better for research and for clinical trials.
Well, it's a pleasure to be here this morning. I do want to be sure, we've been hearing this term biomarkers a lot. I want to be sure you understand what we mean by it. So you heard Dr. Siemers talk about the amyloid plaques in the brain and the tau tangles in the brain. And when you look at the brains of people who have died and you want to make an absolute certain diagnosis of the disease, you look for these plaques and tangles. And as you heard, we know that one includes amyloid, the other includes tau. Well, a biomarker is something that will tell us indirectly what's happening in the brain. You heard Dr. Fillit talk about the fact that we don't tend to do brain biopsies in order to get brain tissue to be certain, so we need some way of identifying the pathology. And that's why biomarkers are so important. We're looking for ways of measuring this amyloid, measuring the tau, or measuring the things that are related to it. So for instance, you all know that the major symptom of Alzheimer's disease is a decline in memory. So we sometimes look at the part of the brain that has to do with memory with magnetic resonance imaging scans. So they tell us not about these proteins, but about the shrinkage of the brain that's happening in the part that's most affected by it. And then as we learn more about the disease, you heard Dr. Siemers refer to the fact that things like inflammation are so important, and people are looking now for measures of inflammation that might be directly related to the disease. So the key is to find these measures that tell us that the pathology is there, and then to use that to make better diagnoses, to make better clinical trials, and to learn in general more about the disease. Now, Dr. Fillit mentioned that you can now, if you think something's wrong with your memory, you go to a physician, they tell you that you do have something wrong with your memory, but they're not sure what's the cause of it. You can go and get a positron emission tomography scan that will tell you if amyloid is present. It won't tell you if tau, I mean, there are scans now that can measure tau, but those are not approved for clinical use. But you can get a PET scan, but as Dr. Fillit mentioned, it's not paid for, so it tends to be very expensive. So we do often do that, particularly in cases that we're not sure of, where the clinical presentation is very, very complicated. So that is certainly one way in which we can use these biomarkers to help with diagnosis. But the really critical purpose for them now, I think, as you've been hearing, is about improving our ability to get better drugs. So in these studies that Dr. Siemers was talking about that recently failed, we know by the virtue of the way the studies were conducted that all the people in the trial had amyloid in their brain because they were scanned with these PET scans. We know that they had trouble with their memory and that they had amyloid. We don't know about other pathologies that they had. So we know as people get older that they're more likely to have combinations of other pathologies, and that's one of the challenges that we have. But the key is that we now can be pretty sure that in these trials are people who have trouble with their memory that's gotten progressively worse, and they have amyloid present. So if you're testing a drug that is trying to remove amyloid or reduce amyloid, of course you want to know that the people in the study have amyloid in their brain. So that's one of the keys. You can also use biomarkers to see whether or not what you're doing makes a difference. Dr. Siemers used the term target engagement. So in addition to finding the right dose to be sure that you're changing, for example, the level of amyloid, you can follow people over time in the trial with various biomarkers. Now, sometimes people do that by repeating these PET scans, but sometimes they do it by repeating the MRI scans, because you might be looking to see if the part of the brain that has to do with memory is not shrinking as much over time as you're treating people. And then, of course, you really want to be able to identify the people who are likely to progress within the course of the clinical trial. And that's actually not something that we've done very successfully yet. So one of the things that I'm working on in my own research has to do with studying people who are normal and trying to find out who's at risk. And one of the keys that we see is that we have to put biomarkers together. We have to measure several of them in order to see that people are going to change within 3 or 4 years. So you might, as you heard, we now know that you can have amyloid and tau in your brain for decades, and you're not necessarily going to change. In a clinical trial, we have to be sure that something's going to happen to the people in the trial within the period of the study. And if we can't enroll people that way, then maybe there are people, they all have the disease, but not enough of it to progress. So that's one of the key ways in which we can use biomarkers that I think is on the horizon. And then lastly, I just allude to the fact, I'm sure we'll come back to it later, we're all aware of the fact that Alzheimer pathology begins to develop in middle age. We know that the amyloid plaques develop. We know that the tau tangles develop. And it's among people who have no symptoms. We think they don't have symptoms because there's not enough of the pathology to kill the nerve cells. But learning about that early phase, tracking people over time, beginning to see how things change is going to be the key. And biomarkers are absolutely critical for that. So I think I'll stop there.
Basically, just to sum up, the MRI exam is important because we can see neurodegeneration, we can see shrinkage. So if somebody comes into the office and they have memory problems, we do cognitive testing to document whether they're off the curve of normal aging. And then we might do blood tests to rule out things like thyroid disease or B12 deficiency. And very often, at least in my practice, and I think most of us will do an MRI exam with volumetric analysis, which is pretty widely clinically available, to see if there's brain shrinkage, which gives us evidence of neurodegeneration. And then perhaps in the future, maybe today for those who can afford it, we can actually do the PET scan that you mentioned to see if people actually have amyloid. And there are other amazing neuroimaging tests like functional MRI or things like PET scans, FDG PET scans that measure function of the brain that can be used. So neuroimaging's really revolutionized the way we can do a differential diagnosis and make an accurate diagnosis. And I think one of the other things that we haven't mentioned was in the Biogen trial that we heard was just a tremendous failure. The drug actually worked. Using the PET amyloid imaging, the drug was there to remove the plaques from the brain. And 80% of the people that got the drug after 12, I forget if it was 12 or 18 months, 80% of the people had no amyloid left in their brain. And you probably would have a hard time making the diagnosis of Alzheimer's in those people. So the drug actually worked, but for whatever reason, in that particular antibody, we didn't see a clinical effect on cognition. But it was a great advance that we could actually use that biomarker as a monitor of the efficacy of the drug and showed for the first time, not necessarily the first time, but in a large study that the drug actually worked, and it's an example of that. I just wanted to ask you, Marilyn, what about cognition? I mean, you know, when we were in the office, we're doing brief cognitive tests, the mini-mental or the MoCA. Could you explain to people what these brief cognitive tests are? Should we be screening people at 65 or 75 for cognitive impairment? And how is cognition being used as a biomarker? Because it seems like that's the real biomarker of Alzheimer's disease.
Well, I'm a neuropsychologist, so I completely agree with you. I'm in favor of having people, when they go for their physical exam, having some simple cognitive testing. Not because you're worried, but because at the end of the day, what we're interested in is change. As people get older, there are lots of changes that are normal. You can be aware of the fact that maybe you're having more trouble coming up with names, or you're not as fast as thinking through problems, but a lot of that is within the normal range. But if you have cognitive testing over time, the physician can see whether or not there's been enough decline that it should be of concern. So I strongly recommend it. Certainly within our own work, we see that this cognitive testing is critically important. So I mentioned that I'm following a group of people who are cognitively normal. They've been in this study since they were in middle age. The average age was 57 when they were enrolled. And we can see now that the tests that they had 20 years ago were good predictors of the fact that 20 years later, or 10 years later, they were going to develop changes in memory. Now, the problem is that that test in and of itself is not good enough. It has to be combined with other information in order to be accurate on an individual basis, but certainly the cognitive testing is critically valuable and important.
Okay, thank you. Our third panelist is Dr. Richard Isaacson, and he's one of the world's leading experts on prevention, and particularly on translating prevention into clinical practice. He's the director of the Alzheimer's Prevention Clinic at Weill Cornell Memory Disorders and an associate professor and assistant dean at the Weill Cornell Medical School, and has a personal interest in Alzheimer's that's related to his family and has helped him to dedicate his career to Alzheimer's research, and he's going to talk to us about prevention of Alzheimer's disease.
Sure. Well, thanks so much for having me. I apologize. I didn't bring talking points. I'm not prepared. You invited me, Harold. There's a fashion show. I thought I was walking on the runway or something, but I wore my skinny-fitting suit and the Donald J. Pliner shoes. But Howard, you're killing me. You put me on the spot here. So Alzheimer's is obviously not a laughing matter, but it's personal to all of us. And what we're trying to do is we're trying to shift the focus of Alzheimer's disease earlier. So we've heard that Alzheimer's starts in the brain decades before symptoms. We even go further than that. We started an Alzheimer's clinic. It's really a prevention clinic or risk reduction clinic, whichever words you'd like to use. And we see patients age 25 and up. Okay, so it's a little different. We'll see a person with Alzheimer's, and then we'll see their family members. And we'll see the kids. I have one family. We see 6 people in the family, 2 people with Alzheimer's, and then we see a first cousin, and we see 2 of the children, and then we see an uncle who's not affected. So we've taken quite a different approach, and we've really used the building blocks that the experts to my right, your left, have really brought to the field. And we do comprehensive cognitive tests. We do computer-based testing to try to figure out, is there something that we can detect early on? And Dr. Albert, you brought this to the surface, and then we're trying to translate this in clinical practice. So we see patients. It's a clinic. I'm an Alzheimer's prevention neurologist. You probably haven't heard that term before, but hopefully you will hear this term much more frequently. There's not a doctor that a person can go to today in most cities where if you want to talk about how do I reduce my risk, is there anything that I can do? Is there anything I can do at all to potentially reduce my risk that's safe generally and has some degree of evidence? There's just not enough doctors out there that you can go to. Well, we were one of the first clinics. There's now several throughout the United States, but we see people across all age ranges. We look at biomarkers that may not be traditionally thought of as an Alzheimer's biomarker. So we call it the ABCs of Alzheimer's risk reduction. So A is for anthropometrics. What exactly is that word? It's a fancy word for body composition. What is a person's body fat? What is a person's muscle mass? As the belly size gets bigger, the memory center in the brain gets smaller. Okay? Interesting. So I was at 8 o'clock this morning. I was in DuPont Circle going to my flywheel spin class, trying to get my belly size to fit in my skinny suit. So I thought this was a fashion show, but anyway. So these are the conceptual things. We're trying to take clinical measures that anyone can check to try to correlate, is there a problem with memory that may happen later? Okay? So that's the A. The B is blood-based biomarkers, and we're hopefully going to have a specific test soon, but we look at markers of cholesterol, markers of inflammation, markers of metabolism. What is the fasting blood sugar? You may have heard of something called the hemoglobin A1C. You've probably heard of vitamin levels and omega-3 levels. Well, we look at all of these markers, and we also look at genetics. And we take these markers and we personalize, or we craft a targeted intervention plan for each person. There's no one-size-fits-all approach towards risk reduction for Alzheimer's. We use biomarkers to inform our care. So if a person has an ApoE4 gene, well, maybe we need to do something different for that person compared to if they don't. If a person has high cholesterol, well, not so fast. Is it high HDL, the good cholesterol, or is it high LDL, the bad cholesterol? So we take all these different pieces of the puzzle, and we try to give a very specific, targeted, crafted plan. And the term we use is based on clinical precision medicine, okay? This is an NIH worded term where we use a specific amount of information about a patient to give specific therapies to a person. And then finally, the C in the ABC is among the most important, and I didn't realize this 5-6 years ago, but that's cognition. We look at memory function. We look at learning ability. We look at processing speed and attention, and we look at higher order function called executive function, okay? We look at all these measures, and someone who's 25, we're doing cognitive tests. You think we're going to find something? Believe it or not, sometimes we may and sometimes we do. Lower than expected, suboptimal, can still be normal, but it's lower than expected. So setting a baseline early in life, following people over time, and then also what we're using is digital biomarkers. So I made a little eye contact over here. I showed you guys what I wear. In my program, we have a little team. We wear biosensors, wrist biosensors, and we can track activity levels. We can track exercise strain. We can track sleep patterns. So we're trying to collect as much data as possible to look for signals in a real-life clinical setting. Also, we collect writing samples. So you may have heard of a study called the Nun study. Nuns in their 20s, when they wrote an autobiographical sketch, and then you looked at the writing samples 60 years later, you could predict who was going to get dementia from their writing sample in their 20s. Wow. So this is biomarkers. We've said that Alzheimer's disease is a 20, 30-year disease. I believe it's potentially longer than that. It's a disease of early and middle age. And as another example, if you're born with a specific Alzheimer's gene, in utero, and when the baby is born, the brain is smaller. Okay? So by taking a different vein of looking at Alzheimer's in a little bit of a different way, we can take the scientific advances of my colleagues here and apply them as early as possible in an effort towards prevention.
Thank you, Richard. Let me ask you, you have outcomes data actually, don't you? Because you're actually not just kind of practicing out there in the wind, but you're actually doing academic work. Yeah. Could you tell us about some of your outcomes that you've gotten from this early, I know you've only been doing this a few years now, but you already have some outcomes data.
Yep, so we follow a cohort. We've seen just over 600 people since 2013. About half of the cohort, a little less, already has dementia, so mild, moderate, and severe dementia. And those patients are in one cohort. And then we have a prevention cohort that includes people with no cognitive trouble at all and normal brains. We also have people in the cohort, or a group of patients, that have amyloid in the brain, but no symptoms. That's called preclinical or pre-symptomatic stage 1 Alzheimer's disease. You don't usually think of Alzheimer's disease as a person having no symptoms. We have people in their 40s, several, 50s, 60s, and earlier that have Alzheimer's in their brain but no symptoms yet. So we follow those people. And then we also have another group where we follow people that have the earliest phases of Alzheimer's disease dementia, but it's pre-dementia, and that's called mild cognitive impairment due to Alzheimer's disease. And then we have people that are just on the cusp of dementia, and that's what we call early mild dementia due to Alzheimer's disease. So we follow these patients, and we have just over 300 or so patients we've been following now for several years. And we'll be presenting the first of our results, finally. It's taken a little while, at the American Academy of Neurology meeting in Philadelphia in a few weeks. And then we're presenting the whole mashbuka, so to speak. We're presenting everything, literally every bit of data at the AAIC meeting in Los Angeles on July 17th at 2:45 p.m. Not like I'm counting or anything. But basically, we've been following patients, and we look at their baseline cognition, cognitive function, their baseline risk, both Alzheimer's and cardiovascular risk, and their baseline blood biomarkers of risk. And we're looking at baseline versus 18 months. That was our primary outcome. We posted it on clinicaltrials.gov, so we have to, stick to the 18-month thing. But we are following people for much longer than that. And I can give you a little bit of a sneak peek, but the moral of the story is the earlier you start with targeted risk reduction. So these are patients that we followed every 6 months. And from baseline to 18 months, on average, people were given 21 different recommendations. Now, Mrs. Smith may have gotten 27 recommendations. Mr. Jones, different, younger, less risk factors, he may have gotten 18 recommendations. Those recommendations are different per person. Again, targeted, precision medicine. But the moral of the story is from baseline to 18 months, the data will be showing in just about a week or so, is that the earlier you go in the spectrum, the prevention cases actually do better than people who already have symptoms. That's one interesting finding. The other aspect is that from baseline to 18 months in the prevention group, you actually see optimization of cognitive function. That's strange. I wasn't exactly expecting that. Now, the complexity here is that we don't have a traditional control group. Control group means half the people get a placebo or a sugar pill and do nothing, and half the people get the multimodal 20-step intervention. Because of ethics, we can't really withhold care. We're a clinic, I'm a doctor, I need to provide good care. So we use two large groups of people, 38,000 people in a group called the National Alzheimer's Coordinating Center that have been taking the same cognitive tests for a long period of time. And we also use the RUSH cohort, which is the NUN study, and in Chicago, where we have 3,600 people or so that have taken the same tests over time. So while our science isn't exactly perfect, because it's not a randomized controlled trial, we do something called comparative effectiveness, pragmatic, real-world research. Taking a step back, we use clinic patients, and we compare them to people that have been taking the same cognitive tests over time, but not getting interventions. Does that kind of make sense? Yeah. And from baseline to 18 months, believe it or not, whether you have high compliance to the interventions or low compliance, the prevention group, the asymptomatic group, actually have been shown to optimize cognitive function. There's actually a statistically significant improvement when compared to the historically matched controls. When it comes to mild cognitive impairment, the data isn't as strong because they're already symptomatic, but only the people that have the highest rate of compliance tend to have a significant benefit.
So I want to wrap it up so we can open up to the audience, but I do have one more question for you, Richard. I know we're helping you to do the Alzheimer's University, where you're trying to teach doctors how to replicate this because you're one clinic in one medical center. How do we disseminate this? How are you teaching other doctors to do prevention for Alzheimer's disease?
Great. So we've created a free resource, and thanks to the ADDF, thank you so much. You've helped us really build and maintain this site. And it's 4 letters, alzu.org. Alzheimer's Universe. It's free coursework. There's 6 different courses. There's a course on what is and what is not in our control for prevention, our prevention course. That's free for the public. There's 12 lessons now, thanks to Howard. We actually have several lessons that were sponsored by the ADDF, alzu.org, totally free. You can go online, you can do some cognitive assessments every 6 months, you can take a course. It's interactive media, it's voice and video and clicking and dragging and dropping. We have courses for high school students, for college students. Told you we go early, trying to educate about brain health. And then we also just launched a course for healthcare providers. So we have a CME accredited course. So a doctor needs to renew their license. They need to get CME or continuing credit. And just since December, we've had just over 400 physicians all around the country and all around the world. Over about 150 have completed the whole course. These are new data, hot off the press. So we're trying to educate the public. We're trying to educate physicians about this topic. And we've, as of a few weeks ago, we've reached 1.25 million people on the site in the last 5 years.
And I just want to say that the ADDF has a website called cognitivevitality.org, which has information from our scientific experts, PhD neuroscientists on staff, that's evaluating the value of supplements and other interventions for Alzheimer's disease. And last year we had over 500,000 people access that website asking questions about DHA and L-serine and a whole bunch of other types of supplements with credible and understandable scientific evidence backing the reports that we've put out there. We have over 150 reports on the website. That's cognitivevitality.org, and it's also available on Richard's website. I want to open it up to questions now. Anybody have any questions? Yes, ma'am.
Having been to some of these wonderful sessions before, there was some suspicion that Parkinson's disease had some correlation with Alzheimer's. Is there any update on that? And a quick second, which is, I'm so thrilled to hear that 57 is middle age. But for those of us who have had that Medicare cognitive test, it's actually pretty silly. And I wonder how we can improve that, or at least make it a little more sophisticated than what day of the week is it. Thank you.
Yeah, well, maybe I'll take a first crack. There are different kinds of dementia. Parkinson's can be associated with dementia. The pathology is different. It's called Lewy body pathology. About 35% of people with Alzheimer's pathology also have Lewy body pathology. So what we've learned in the last few years actually is that these diseases are not as pure as we thought about. We probably have continuums along the line, and that most people probably suffer from multiple comorbid pathologies, which tells us that these clinical trials are going to be much more difficult than we originally thought. But definitely people with, about 20% of people with Parkinson's will become demented. The difference is that they usually develop their dementia late or later in the course of their Parkinson's disease, so they're going to get motor symptoms first, and then along the way later on they'll become cognitively impaired. And as far as the mental status is, and I'm going to turn this to the panel, but I just want to say that the answer is that these screening instruments are there to detect significant clinical impairment. So for the normal person, you're absolutely right, there's what we call a ceiling effect, that they're not sensitive enough. And that's why Marilyn was saying that we need better tests for your population, and it's difficult. So I'm going to ask Marilyn to address the neuropsychological component of this.
Well, by and large, we know specifically for Alzheimer's disease that most people start with memory problems. And so the evidence suggests that really good memory tests are going to be the most sensitive to early changes. But I would emphasize that these simple tests, yes, they have a ceiling effect, but when you begin to see any change at all, it's probably relevant. And it's the best we can do right now to get into a medical setting, but obviously we want to do more. And the kinds of tools people are trying to develop now are things on computers that you could easily have in a medical office. Somebody could maybe sit and do it while you're waiting, and that would be clearly in advance.
And the development of tests and their implication, introduction into clinical practice is difficult because a lot of people don't want to know. And some people do. And so in practice, it's really kind of almost in the art of medicine. If somebody comes into my office and really wants to know, and they're kind of normal, and they have what we've defined as subjective memory impairment. So on those tests that you talked about, they're not sensitive enough. Let's say somebody is highly intelligent, and they were, highly intelligent, lots of years of education, and now they're 57, and they're down to here, and they know it, and family members know it. And maybe some of the problems are in what we call executive function, abstract reasoning, multitasking, judgment, emotional control, these kinds of tests. We don't really have good neuropsychological tests for those kinds of issues, which may be very early. And so there's a clinical issue here. Under Medicare, we have the wellness exam, which is paid for annually by Medicare. And within that, the law states that there should be cognitive testing. But something like only 5% of people actually get the cognitive testing. Either doctors don't have time for it, they're not bothered with it, they don't want to make a diagnosis because it takes a lot of time to then counsel people and go through a diagnostic workup. And so even today, most people with Alzheimer's disease don't get actually diagnosed until they're in the moderate or mild to moderate stages of dementia, which is a terrible thing because it leads to unnecessary hospitalizations and falls and all kinds of caregiver stress. So the issue you're raising about early diagnosis is really important. But the future of it, I think, is very good because even those people with subjective memory impairment, we know that people with subjective memory impairment, where the only detection we can see is the person telling us or the loved one telling us that there's something going on, but we don't really document it. But people with that who also have biomarker evidence of Alzheimer's disease are the ones who are going to progress. So there's a clinical model that we see in the future. And when the blood test comes available, those people like yourself who have this subjective memory impairment that's not documented by neuropsych will be able to get a blood test, and that will put them on a more traditional diagnostic path. Any other questions? Yes.
Yeah, is it possible that somebody has been diagnosed with Alzheimer's dementia, middle age, that they actually the brain's not allowing them to recognize that they're forgetting or something is wrong, even if the doctor looked them in the, yes.
So,
Talk about an SNO
It's
here.
not that they're in
Well,
denial. It's
so
not
now-
that they heard it.
That's right.
So what I've been doing for a really long time is following people over time. And in this study that I was mentioning now, we follow people from the time they're middle age. Every year we see them. And what you see happening when people begin to have trouble, mild cognitive impairment, very mild symptoms, is that they're aware of the symptoms. They complain to you by and large. They see that there are problems. You can ask them clearly, are you repeating yourself more? Are you forgetting conversations? Are you having more trouble getting your groceries? All of these kinds of simple questions, people give you answers that are accurate. Once people begin to get more impaired and develop dementia, so that's to the point where they're unable to manage on their own, many of them lose that awareness. So they lose insight into what's happening. And it used to be said that if you thought you had Alzheimer's disease, you didn't. So that isn't true, because some people do retain that insight, but you can see people when you follow them over time, that then one year, all of a sudden they say, oh, I'm really not having so much trouble anymore, when in fact their family members are saying they're having a lot of trouble.
And
so it's a change in the brain that's altering, we think, the frontal lobes of the brain, that's making it hard for people to then recognize the symptoms that they have.
Yes. Any other questions? Yes, in the back.
There are many children in this country who are on stimulants. Has that been studied at all vis-a-vis future issues, brain issues?
Anybody?
Yeah, I get this question quite a bit. I wouldn't say that I have the best available evidence to make one way or the other, but I haven't seen convincing evidence that stimulants will lead to something later. I can say that from our cohort, people who have learning disabilities, or dyslexia, for example, or have attention deficit disorder, do present differently. And these are people in their 20s, 30s, 40s, 50s, and beyond. Their cognitive testing is different, their executive function is different, and they may present actually with atypical Alzheimer's. So I wouldn't say that these drugs increase risk, to my knowledge, but I would say that people that are treated with these drugs may present differently later in life.
And I would just add that at our institution, there are actually some clinical trials using stimulants in people who have Alzheimer's dementia, but are very apathetic and aren't active and not engaged and not involved, and trying to see whether or not this can improve their function, not prevent the disease, but just improve their day-to-day function.
And that's an important point, because 90% of people with mild cognitive impairment and early dementia have apathy. And we think of apathy as a generic term, but it's actually a very important neurological symptom, because people, in our terms in dementia, apathy means that people may have the desire to do something, but they can't execute it, or they don't even have those desires. And Marilyn, maybe you can define apathy from a neurological point of view better than I can.
Well, I mean, it's just that people tend not to initiate, they tend not to, you hear from families all the time, they used to be interested in something, they used to go and be with their friends, they used to go out to dinner, they used to want to see the movies. It's that lack of interest and initiative. And if you could improve that, you could improve people's daily function, and that's what these clinical trials are about.
Any other?
Maybe just one other quick comment that relates to actually the last two questions. One of the shifts in the field is how you define the disease. You know, the question came up, if somebody doesn't acknowledge it, what does that mean? So as Howard pointed out, you know, back in the old days, it would take, well, an autopsy. It was actually the gold standard, or a brain biopsy. But now that we have these biomarkers, we can tell you if you have the amyloid plaques in your brain to a great extent. And that sort of biomarker evidence of the plaques and also these tangles, which is still, they're not quite available yet. But once those are available, that's really how you define the disease. The symptoms can be different for different people, but the disease is actually defined by the pathology, which is what most of medicine does. Now, the most controversial part of that has been, well, if you have the pathology but you're cognitively normal, is that really a disease? It's one of those, if a tree falls in a forest questions. I think there's a lot of people that would agree that if you're really talking about prevention, you want to be aware of that pathology even in a person who doesn't have symptoms. But that's been a real shift in the field that the NIH and the Alzheimer's Association put together a working group, and the paper that resulted really said, we're going to define this disease by the pathology because the symptoms can vary so much from person to person, and especially depending on what stage of the disease you're in.
And the FDA changed their guidelines to include biomarkers as definitions of the disease that could be treatable. And the other thing I think we're talking about is more symptomatic treatments for things like apathy, for agitation, which affects up to 90% of people over the course of the illness and is a major risk factor for institutionalization. ADDF has funded a couple of clinical trials of new drugs for agitation because right now the only drugs that are available for people with agitation have what the FDA calls a black box warning. And it says on the front page of the FDA material for these drugs that these drugs are putting elderly people with dementia at risk for death. So as a practicing physician, somebody calls me and says, or I see a patient who's agitated, is up all night, might be physically or verbally abusive, they're out of control, and patient says, the loved one says, what can I do? And I can say, well, I can prescribe you this drug, but it has a black box warning, and puts everyone at risk. So they're not very effective drugs. So developing new drugs for some of the alternate symptoms of Alzheimer's that have been raised is really important. Any other questions? Yes?
Just to follow up on that, question about the amyloid presentation. With regard to the amyloid presentation but no symptoms, is it ever the case that you never develop symptoms, you just have the amyloid?
Yeah, so great question. There are many people in the field would say that if you live long enough, you probably will develop symptoms. But if you get hit by a car the next day, then you obviously wouldn't. But the practical point, and this is, and it gets a little bit to what Dr. Albert talked about too, is we need to find predictors of who will have a decline. So again, you have the plaques 15 to 20 years before you have symptoms. So some people will become symptomatic, but not everyone, and we need to be able to sort out who is really at risk for developing symptoms.
And there are blood tests now that are being researched that the Diagnostic Accelerator will be supporting that will be able to predict, or seem to be able to predict, who is going to become symptomatic. Tests like neurofilament light, which is a test that's being used in multiple sclerosis and so on, that actually, along with the other diagnostic tests, seems to predict, as a real measure of neurodegeneration in the brain, who might go on to decline. So I think the future is going to be better predictors of using blood tests, and also new kinds of brain scans will have better predictive ability. And part of the reason is that although amyloid is a very good diagnostic marker, the presence of amyloid or the amount of amyloid load doesn't really correlate very well with cognitive impairment. The Tangle tests that we're going to have on the market probably in 2 or 3 years is a much better predictor because the tangles are the dying neurons themselves. But the most sensitive marker is not the dying neurons, it's the synaptic connections, the trillions of synaptic connections in the brain. And we actually even have brain scans right now that can look, a test called SV2A, that can actually even look at the synaptic connections on brain scanning. There's another blood test that's being developed that's already in the spinal fluid called neurogranin. That's a measure of synaptic synaptic dysfunction. It's starting to be used in clinical trials. So the future, I think, is very bright for us being able to predict better. And as Richard mentioned, there's this apolipoprotein E test that anybody can get today. You can go to Quest or LabCorp or whatever and see if you have apolipoprotein E4. 20% of the people in the community have an E4 gene. Get one from mom, one from dad. If you have both, you're E4/4, your risk of getting Alzheimer's clinical symptoms is about 5 or 10 or 15 times, about 15 times greater. And if you only have one, it's about 5 times greater. And 20% of people in the community have E4, and about 60% of people that suffer dementia from Alzheimer's have an E4. So we're gaining that knowledge, and it's going to be translated into clinical practice, I think, in the next few years.
Here is my actual question. That was just like a follow-up from what you guys were talking about a minute ago. My actual question was for Dr. Siemers about the clinical trials about the immune system. Can you talk about that a little bit more? What is it about the immune system and Alzheimer's that makes you want to study it?
Right. Well, for years, people have looked at brains with people with Alzheimer's under the microscope and seen these little cells called microglia that are the sort of like it's like a white blood cell in your brain, and typically those are going to fight off infections, but you also see them around the amyloid plaques and things. So there's been this long idea that maybe there was too much inflammation there, but it wasn't very clear whether those were really causing bad effects or not, and some of the early studies with anti-inflammatories didn't work, and so it was controversial, but some of the more recent genetic studies, again, now start to point to different places in the immune system, and it's probably like there's not either too much or too little immune response. The immune response is really a complicated thing, and it'll probably need to be dissected out to some degree. So one of the genes that was found is a gene called TREM2, and just to give you an example, and so that gene, the gene then makes a protein, and then there's a drug that just entered clinical trials that acts at that TREM2 protein. So these are early days, and that would be to dampen down inflammation. But these are early days, but there are good targets out there, and we're just going to have to dissect out which ones show a clinical effect.
And another way that works, I think I might add, is not just brain inflammation, but the effect of systemic inflammation on the brain that's activating these microglia. So, for example, if anybody's ever been sick, we know that there's a syndrome called sickness behavior, which is the effect of systemic inflammation on the brain. So, for example, in geriatric medicine, we teach that we all know that somebody that has pneumonia, who's 30, 40, they have cough and fever. But an 80-year-old, the most common symptom of pneumonia that we look out for is confusion, and it's the effect of systemic inflammation on the brain. So there's an interaction there between aging and systemic inflammation, which is a hallmark of aging, and the activation of neuroinflammation, which would be then a different kind of target. So we have two kinds of targets, and we've run some clinical trials, and some of them in the past to try to reduce the systemic inflammation to see if it'll have a positive effect. We have time for one more question. I'm sorry to ask you right there.
Can you give some examples of the 18 targeted interventions that you give to this patient and the 23 that you give to this patient?
Sure. So the question is, can you give some examples of the targeted interventions? So if you go online, you can learn all about this. So I'll give you a short course right now. But the general categories are education. So for example, say a 59-year-old perimenopausal woman comes in, she's on hormone replacement, should she be on it or not? She has increased abdominal obesity, meaning the belly gets bigger. She may have higher than expected fasting blood sugar. She may be physically active, meaning I walk 20 minutes 5 times a week. Well, there's a difference between physical activity and physical exercise, okay? And maybe when we do the memory testing, she's normal, but she has lower than expected for her age and education. So someone like that is on a metabolic pathway, potentially. She has one copy of the ApoE4 gene, as an example. So we take all these different tidbits, and for example, we'll talk about education first. Hormone replacement therapy. How long should you be on it for? What are you on? Are you on a pill? Are you on a transdermal? Do you have symptoms? Why were you put on it? How many years? So that's number one. Number two, we'll talk about metabolism and exercise and diet. There's no one size fits all to a perfect brain healthy diet, but this person should probably be on a Mediterranean style diet, maybe with a little bit carbohydrate restriction, okay? So maybe even amp it up a little more, maybe some carbohydrate restriction for 12 to 14 hours overnight to calm down the insulin resistance that she's having. Maybe she needs to have more intense exercise, twice a week. High intensity interval training can ramp up the mitochondria. A recent study showed that high intensity interval training can even help with menopausal, perimenopausal symptoms. So we'll give targeted advice about exercise, targeted about diet. She also loves chocolate, and she eats chocolate bars, but chocolate bars have sugar and butter and more sugar and butter. So we'll give purified dark cocoa flavanols, which has been studied in multiple randomized trials, purified cocoa powder, which can improve memory function, improve blood pressure control, and insulin resistance. So we'll give a supplement that's a cocoa flavanol, and we'll look at her markers. She has an ApoE4 gene, so she may be preferentially likely to respond to omega-3 fatty acids. Well, she also is low on omega-3s and doesn't eat fish. So I'll say, hey, why don't you try sardines, wild salmon, mackerel, herring, albacore tuna, at least once, twice a week. And since your levels are low, and I don't know that you're really going to eat fish, maybe you're the type of person that should be on omega-3s, but which omega-3s needs to be DHA and EPA, and we'll titrate that up based on her cholesterol level, because her LDL was high, and the particle size was even higher, so we need to calm that down and give more omega-3s. We're also going to talk about stress reduction and sleep and all sorts of other things, and those are her 18 steps.
Well, I want to thank our terrific panel for their participation and thank all of you.